quite an international affair with both some industry collaborators as well as academic partners. And also thank you for funding from the NIH and the Department of Energy for supporting both of these projects, or supporting this current project. Oops, let me make sure I can advance the slides. There we go. So I don't think much needs to be said, but in general, we're all quite aware that there is a significant push for this development of novel quantum sensing applications, and in general, like this kind of environment of quantum computing and quantum communications, quantum sensing as a broad field in general has shown significant growth over the past decade, well, 2 decades or so, with really the promise of being able to measure interactions that especially on the quantum sensing side, at extremely small length scales with very high sensitivity and resolution. So I just highlight some general exciting on the left, just some recent publications, as well as a nice, really excellent review on the just general concept of quantum sensing from Paolo Capolero and collaborators from 2017. On the bottom right is actually just from a sort of investment management group assessment of this quantum, in general, quantum technology in general, which includes encompassing quantum computing, communications, and quantum sensing. So there's a lot of anticipated in growth over the next decade or so expected. Okay. All right. So amongst all of those broad materials of quantum sensing, which includes NV and things like two-dimensional materials, here, of course, our focus is on NV, and NV and diamond has been utilized for free radical sensing in a variety of temperature sensing, nanoscale magnetometry, and a lot of these are utilizing these kind of exquisite coherence and spin properties of the nitrogen vacancy center, which I'm sure many of you are aware of. So the ability to initialize and optically read out spin states at room temperature with the NV center is really an exceptional piece of materials science that allows us to get really the motivating factor for a lot of these quantum sensing applications. Okay, so in general, we want to explore like why it's important to reduce the nitrogen content. So as a general overview, what I want to talk about today is why it's important to reduce the nitrogen content in the context of quantum sensing. How can we control the nitrogen content, and specifically with HPHT synthetic diamond. So there's significant advancements in CVD diamond that have happened over the past decade, but HPHT is an older technology, but is more suitable for particle synthesis. We'll just briefly review what the current state of the commercially available diamond and the nitrogen contents in those, and then I will go into our the material of my specific interest, which is a series of microparticles synthesized with 3 levels of nitrogen content. Okay. So why do we need to control the nitrogen content? So from that review, and in general, it's the sensitivity, which is the minimum detectable signal as it's defined in this community, is generally inversely related to a square of a coherence time or a phase memory time to T2 star, and these spin coherence times, as you can see here, the relaxation rate are generally proportional to the concentrations of the spins, and then typically in a dilute spin bath like in diamond, you have these dipolar interactions which are dominating the relaxation rates. Okay, so in this case, in particular, nitrogen in diamond forms a paramagnetic P1 center, which are well established to be a major source of decoherence for NV centers in bulk diamonds. So you can see on the bottom right the function, and these are in bulk crystals. So the effect of nitrogen concentration on the coherence time. Okay. And why do you, and of course you get better sensitivity for an enhanced coherence time. Okay. I highlight 3 papers on the bottom left. So these are examples a fairly, well, at least the top one is a fairly recent example where CVD diamond was milled, and they observed like very high coherence times in those milled nanodiamonds. A disadvantage of CVD is really the cost. And then the bottom two are sort of related papers where the claim was a source of HPHT diamond actually in the Nature Materials paper, and then the subsequent nanoscale NMR imaging nanoscale paper was that the nitrogen content being reduced to approximately 30 ppm really enabled their use in this nanoscale NMR due to the extended coherence times. Okay. So there's a great interest in if we can really control. So the disadvantage of a limitation of those papers is that they typically buy material from a producer, and you don't actually know how reproducible such this material is. So what we're looking at in our study is straight from the producer, from the starting mother crystals, right? Mill, and we will eventually get to the point of milling it down. Okay. So how can we actually control the nitrogen content in HPHT synthesis? So in general, this involves the introduction of some nitrogen getters, which simply just compete, maybe forming some nitrides in the melt inside of the synthesis chamber. So an example of these, it might be aluminum. You can also enrich the nitrogen content. In general, I would say for quantum sensing applications, it's not maybe not the direction to go, but I just highlight it here to show that you can actually regulate the nitrogen content from very high levels of, say, up to like thousands of ppm. And in bulk crystal HPHT, you can grow all the way down to even less than 10 part per billion levels of a substitutional nitrogen. So on par with, say, some of the best CVD as well. Okay. All right, so just to give you a general overview of the commercial current state of the art, and this is mostly bulk diamonds, so you can see CVD and HPHT synthesis allows for the growth of very high purity diamond, even down to the lowest levels of part per billions and part per billion levels of nitrogen. Those tend to be very expensive, of course, because as purity goes up, right, cost goes up. Standard HPHT diamond on the market is typically containing around 100 to 300 ppm of nitrogen, which is shown in yellow. And you can see actually as you increase the nitrogen content, you can shift this color to this greenish. So these are all HPHT synthesized. Currently, there is some emergence of these CVD grown high quality, somewhat intermediate nitrogen content. So the two plates that are shown in the middle, the pinkish plates, so these are these element 6 DNV plates, which have recently come onto the market. So they have sort of a good, reasonable ensemble of NVs, NV minus, which are suitable for many sensing applications. But in general, particles have been limited to the range of, say, 100 to 300 ppm. So, and why would we want to use particles as opposed to plates? Well, in general, particles, if you can get them in good enough quality, they're more easily interfaced with, say, nanoscale environments through chemical functionalization. So we can actually get ideally higher precision, higher spatial precision measurements. A challenge with plates is often like how to interface the NV inside of the bulk plate with a biological analyte of interest. So in principle, particles can offer better ability to do that through various chemical functionalizations as well. Okay. So this is just an overview of the experimental design. So we have essentially three particles, which are approximately 100 micron in size, that were grown via HPHT by our collaborators at Hyperion, which is a commercial industrial diamond producer based in Ohio. So we have three levels of nitrogen content, so approximately 30 to 40 ppm of nitrogen at the highest level, and down to about 10 ppm in the lowest sample. So just to remind you that the typical HPHT particles have about 100 to 300 part per million of nitrogen. So even our highest nitrogen content is lower than typical. We do a variety of characterization to assess both the content of the nitrogen and the general trends in the PL spectra. as well as some single particle ODMR characterization from our collaborators at RMIT in Australia. I do a series of electron paramagnetic resonance experiments, both continuous wave and pulse, to assess relaxation times and spin content. Additionally, we have a glow discharge mass spec for metal content analysis, and our collaborators at Berkeley and the Ashok Ajoy's group doing some NMR carbon-13 transverse lifetime measurements. And the purpose of those measurements in general is the idea of using the carbon-13 in the particles as a sensing element. So if you can polarize NV and then transfer polarization to the carbon-13, the advantage of the carbon-13 are the typically very long nuclear spin relaxation times and coherence times. So you can get even much higher sensitivities. Okay. All right. And I would say, just in terms of irradiation was done in Japan with approximately 2 MeV electrons, which I highlight on the bottom, with a fluence of about 1 × 10⁸ and then subsequently annealed in vacuum at 850 for 2 hours. So in general, pretty modest irradiation conditions, low fluence. So, okay. So if we start with just simple optical characterization of the as-received particles, you can see in the top left that there is a trend from the highest nitrogen content having a slightly more yellow color down to nearly colorless at the lowest levels of nitrogen. So this is generally expected as the nitrogen P1 centers contribute to this yellow coloration of typical HPHT diamonds. So we have sort of a verification here at the beginning that we see a trend of reducing nitrogen content, which can then be supported by more quantitative characterization. On the right, you can see just some PL spectra of some representative particles from each in their as-received state. So as received from Hyperion, and in general, you will see like a reduction in NV, intrinsic NV. So these are NVs which form naturally during synthesis. So you can take essentially any HPHT diamond, and I'm sure if you shine a laser on it, you will find some spurious NV centers in most particles. So it's nice to see here a general trend that with the lowest nitrogen content, you do not see many intrinsic NV centers formed during synthesis. So on the bottom left is the results of this glow discharge mass spec for our mid-nitrogen sample, which has about 20 ppm of nitrogen. So, a general disadvantage of HPHT because of metal catalysts used during synthesis is the incorporation of metals is inevitable. But that as compared to, say, CVD, which you have no metal content, no metal contamination. But the thing to draw your attention to is really that the high aluminum content. So this is probably related to this nitrogen gettering role, and it's incorporating into the particles. Okay. So here is just after irradiation and annealing, some PL characterization. The thing on the top left, you can see just fluorescent images of the particles. And I think it's most apparent under this green excitation, which in this case is around like 520 nanometer. You see a generally blue shifted, especially between the high and the mid end, you see a slight reddish coloration shift to a slightly orange. And this is backed up by, on the top right, the PL spectra of these particles. So you see a generally blue shifted emission intensity. And this is the result of having insufficient charge donors in the lattice, and therefore you form less NV minus and then start to see more NV zero. And in the extreme case on the bottom right of a CVD plate, so these have typically less than 1 part per million starting nitrogen. This has been irradiated with electrons and not at the same facility. But here you see almost exclusively NV zero due to the insufficient charge donors in the lattice. So it's a nice verification here that you're seeing the effect of the nitrogen content on the PL spectra here. And that's an important consideration because we really want NV minus rather than NV zero. But this is a compromise that you have to start to consider with reducing the nitrogen content using traditional synthesis methods of NV centers, which are electron radiation and annealing. So the bottom left figure is just summarizing these results. So you see as the nitrogen content's going down, you see a decrease in NV minus, increase in NV zero. And then I will subsequently show that you have an increase in the NV minus T2 time. Okay. So I'm going to start going through the EPR data, and we'll start with the continuous wave. And in general, we're considered with two signals of interest. So the top left two figures are simulated P1 spectrum, powder spectrums in derivative mode. So, and this is usually the dominant, this is your nitrogen. So this is the dominant signal in most HPHT diamond. And on the bottom left, you will see a powder spectrum for the NV minus, which is a triplet, so S equals 1. And the main signal we're interested in is what's highlighted with this half field transition. So this is our W15 or NV related it's transition, which we use for quantifying the NV content. So in general, the results of the concentrations for as received and irradiated annealed P1 content and NV content are in the bottom left. So you see in general, as expected, you see typically a reduction in the P1 content after irradiation and annealing. And from measurements of the half-field signal and double integration of the EPR signal, we can quantify the NV contents. And the conversion efficiencies are quite low, and it's probably related to, in general, the low, modest conditions of, possibly related to the modest conditions of irradiation and annealing that were done. On the top right, these are the continuous wave P1 spectra for each of the particle samples from high to low. So the thing to note here is the middle nitrogen sample having first of all, a slightly narrower line width. So in general, you would expect that the line width is inversely related to the relaxation rates for a homogeneously broadened line. And on the bottom, you see in the lowest nitrogen content, we see quite a broad, like unexpectedly broad line width. And this can be the result possibly of nitrogen clustering. So if you have some nitrogen clustering, you might have a spin-spin interaction, which ends up broadening the line widths of the EPR spectra. On the bottom right, you see suppression of this NV half-field signal as we go from high to low. So that's all within reasonable what we would expect with lower nitrogen content. So the NV minus production efficiency, well, I don't want to say efficiency, but the amount of NV minus essentially that you get is decreasing, right, with reducing nitrogen content. Okay. So continuing with the continuous wave. So this is what's called a microwave power saturation experiments. So essentially we sweep the microwave power from low to high, and we can monitor the EPR spectra as a function of this microwave power. And if we take the peak-to-peak amplitudes of this P1 central line, these are plotted on the right. So, I mean, what does this information tell you? So just some background on EPR, if you're not familiar. This technique was kind of really before the development of a lot of advanced pulsed EPR spectroscopy. This was kind of the way one would assess relaxation phenomena in materials. So this is quite an old 1960s, 1970s or so. So the important thing is this equation on the bottom. So this is the peak to the amplitude of the derivative signal of a homogeneously broadened Lorentzian line, which and the important thing to take away from this is the saturation term in the bottom right. So notice that as T1 and T2 would increase, this would pull down the derivative amplitude. So this is what's called saturation. As you increase power, you can start to saturate the signal. So let's consider for a moment that you have kind of a fixed power. The longer T1 and T2 are, the easier they are to saturate. So you typically see below saturation, you have this linear increase in the peak-to-peak amplitude. And then at some point, you start to saturate. So if this happens at lower power, it's an indication that the relaxation times are longer. So it's sort of qualitative. Now, historically, people have done some types of models and fits to this. In general, this is very complicated to do because you make some potentially severe assumptions about the spin systems. So in general, I just rely on this as a qualitative measure. So The thing that's quite interesting is in the lowest nitrogen sample in the top right, you see basically it's increasing, and then it starts to saturate, and then suddenly this other signal after radiation annealing. So the dashed line increases. So generally speaking, lines, the lines in diamond and in most solids are called inhomogeneously broadened, and one source of inhomogeneous broadening is overlapping spin systems, right? So you might be saturating P1, but there's some other spin system perhaps related to the irradiation damage, so maybe vacancies, which are then faster relaxing, and then they start to dominate the signal. So perhaps what's happened is our annealing conditions were maybe not sufficient to anneal out irradiation damage in that sample. And the ability to anneal that out might be due to the nitrogen content as well, okay? So in order to assess relaxation times more accurately, we rely on pulse DPR, and this is done at Q-band, so at about 34 gigahertz. So on the left, you can see what are called echo-detected field sweep. Field sweep spectra. So these are the absorption spectra, and you can see the primary signal P1. So just to note, in continuous wave, we typically measure with phase-sensitive detection. So you're going to see like a derivative signal, but here you're just measuring the absorption signal. So you have an echo, which is generated by on the bottom left, so essentially a pi/2 and pi pulse, which generates an echo. And then you integrate under the intensity of this echo to generate the absorption spectrum as a function of field. So that's your how those signals are generated. And on the inset, this is the one of the allowed transitions for the NV, so we can see these. And essentially, I can measure both T1 and T2 by Hahn echo decay for T2 and 3-pulse inversion recovery measurements for T1 measurements, and I can sit on one of the lines to measure those individually. So in the top right corner, you see these Hahn echo decay traces as a function of the nitrogen content. And you see that as the nitrogen content is decreasing, you see an increase in the T2 time. I don't show the NV, the inversion recovery for the NV, but you can see that they're mostly not affected. And it's not maybe surprising because T1 in general does not depend explicitly on spin concentrations, whereas T2 is very much more sensitive to spin concentrations. Okay, so we see almost, roughly almost threefold enhancement of the NVT2 from the highest nitrogen sample to the lowest nitrogen sample. So the other spin component just to look at is the P1. I know everyone cares about NV, of course, but it's also important to monitor the P1 as well. So you can see here in the top left, again, these absorption spectra, and I've highlighted the lines that we're focusing on for our Hahn echo decay and inversion recovery. So again, you see an increase in the P1 coherence time as a function of nitrogen concentration as the nitrogen concentration goes down. In general, the T1 is not affected as much. Maybe at the lowest nitrogen, it's slightly increased. In the top right, so there's a paper from Van Wyck from the 1997 where they were measuring, say, P1 coherence times relaxation rates as a function of nitrogen concentration in a variety of bulk diamonds, and they derived an equation actually using the exact same method. So they're doing pulsed relaxometry measurements in EPR, and you can see they derive this function as the relaxation rate, the inverse spin-spin relaxation rate as a function of the P1 concentration, and I've plotted that there in the red dashed line, and then I've plotted my measured P1 T2 values, relaxation rate values on that plot. So the thing that's nice about that is you see reasonable agreement with that line. So that means that our concentration measurements are reasonably good. Okay. And here, this is the ODMR characterization that has been done by our collaborators at RMIT. So here you can see very different measurement techniques. So AC, DC, AC sensing modality, DC continuous wave. and then both low power pulsed DC and high power pulsed DC. And these are roughly just figures of merit sensitivity. So I don't show the equations here, but there are equations that relate the minimum detectable signal as a function of for these various sensing modalities, which are in the literature. So what we can see is that in general, it seems like the intermediate nitrogen sample is outperforming. So just to clarify again, that the lower value is better sensitivity. So that's actually maybe not immediately expected. You might expect that, oh, the lowest nitrogen is going to give the best result. But the reason is that there is a compromise between, say, brightness and the nitrogen and the coherence times. So if you don't have enough NV, you don't have enough brightness. So you kind of degrade your sensitivity in that way, but by your photon count rate being too low. But in the case of the mid-nitrogen, it seems like an optimal sort of concentration for enhanced sensitivity, at least in this series of particles. So, and again, so this is on the level of about 10 to 20 ppm of nitrogen, which is giving this performance, which is interesting actually because it's about the same that's in the DNV samples. So the CVD samples from element 6, it's about the same order of concentration which they're doing for these ensemble NVs. So it's actually reasonably good agreement with both the CVD diamond as well. Here, I just want to show the tentative data. We're still doing measurements, and so I should add for the previous slide, we're continuing measurements characterization to get more statistical analyses for that. But here, this is some work from Berkeley that's tentative right now, where we're exploiting, just measuring the carbon-13 T2 spin-locked lifetime and the hyperpolarizability, which is another aspect I'm not going to discuss for the moment. But so you see, in general, there is some dependence of the nitrogen content on the T2 relaxation, on the carbon-13 T2 relaxation rate. For the moment, this is still an open question as to what's going on. So possibly at very low nitrogen contents, the polarization transfer is maybe not as, it could potentially be impacted since you don't have enough NV centers, and you might be, well, I don't want to speculate, but that's a possible some reason for that at the moment. Okay. So just to wrap up our general conclusions, can we synthesize good quality HPHT diamond particles and see improvements of NV spin properties by reducing the nitrogen content? And that's even with there's metal contaminants being present. And the answer is, I think, yes. Is the lower nitrogen content better? Yes, with a caveat. So it looks like, there's a compromise, but again, between the brightness and the sensitivity gains by increasing the coherence time. So in this case, we see the best performance in this intermediate nitrogen range. Okay, so then what's next for us? So possibly the NV production efficiency could be improved by increasing the irradiation fluence and some more optimized annealing conditions. Again, we're pretty modest annealing and irradiation and annealing conditions in this work. And then secondly, of course, the really crux of everything is what happens when you crush these to a nanoscale. So there are, of course, these papers which I discussed briefly earlier, which indicate that reducing nitrogen content does offer some benefit even at the nanoscale. So hopefully here we might be able to show this. So this is something that is ongoing work to characterize these particles after crushing to see if there's still a benefit. And again, the nice thing here is that we have it traced all the way back to the mother material, not just all that was already crushed up by the supplier from some particular batch, right? So we're tracing it all the way through. So we have a good, I think, handle on what's happening from that. Okay. So just to wrap up, I'd like to again thank all of the collaborators that have been involved at various universities and some of our industrial collaborators. Yeah. So thanks. Good.
Nicholas, thank you very much for the nice presentation. Now we are open for questions. You can please ask directly or type in chat room. Further, please, probably you have.
Thank you very much.
Nice
Yeah,
to
thank
see
you
you,
very
by the
much,
way.
Nicholas, yeah, for a very nice presentation. I have perhaps two short questions, if I can pose two. So one is regarding your T1 time. So it depends on a nitrogen content, right? Like T1 is shorter for high nitrogen, if I understood correctly.
Sorry, I think the audio cut out. Oh, sorry. You want me to mute?
Yeah,
sorry. Yeah, the audio. Yes, sorry. Can you repeat the question? Sorry.
Yeah. Is T1 shorter for higher nitrogen content? So T1 time?
Yeah, so if I go back to that slide here,
yeah,
just marginally, it looks like, so this is the, like, it's a question of if that difference is significant. I don't know if I can say if that difference is very important, but it's marginally impacting.
Very large, yeah.
Yeah. Much more significant impact on the T2. Yeah.
Okay. And then the second question is regarding your sensitivity estimates per volume. You had this. So is it expected to be normalized on square root of micrometer cube? So it's micrometer square. So I think it should be micrometer.
Yeah. So that's an excellent point. So this is from our collaborators in Australia that are doing these measurements, and they have a wide field microscope, which typically they're measuring in plates. All right, so they normalize the surface area. I agree that perhaps I can maybe a better way to normalize this would be to we're thinking maybe like even just temperature sensitivity. I think the reason they don't scale toward volume is they don't have good I don't want to speak on something that I didn't do, but I think
they don't have a good estimate of volume, but there can be some estimate because the sensitivity should go to the square root of number of NVs. So, so then it's a kind of represent the material. So that's from my perspective.
Okay, so I guess to do that, what I can just well, actually, I would like to
estimate the sense in volume or excitation volume, and then this data can be. So, for example, if your layer is maybe just a few micrometer depth, then you can sort of give here this kind of surface area multiplied by the depth, which will give you a micrometer cube.
Okay. Yeah. I might do that. I have to speak with them. I think one thing that they mentioned is they don't have good Z control of their magnetic field, so maybe that's also an impact. But I can ask them about that. Yeah, it's a good point, because it would make more sense, right? Thank you.
Yeah, thanks.
Please, we are open for additional questions. So, Nicholas, what would be a strategy to reduce metal content, which is undesirable, obviously, in the sample?
It's muted. Yeah, so I think that's going to be potentially tricky. Perhaps like at the synthesis level, they can control things better, which I can't really comment on since I don't have information on their specific conditions of growth. What's possible is that when you crush the diamond down, you might open up surface area that might allow you to then treat an acid to remove some of the metal content. I mean, also on the other hand, these metal relaxation rates, especially at room temperature measurements, might be very fast, so they don't potentially interact maybe with these NV spins as much. So it's kind of like it's like a hummingbird, right, that has flaps that swings very fast, but it doesn't disturb, say the leaf or the environment around it. So it's something like that. At the moment, I don't know a direct means of reducing the metal content in these bulk particles, but perhaps with crushing and treatment. I think it's inevitably just okay. That's the compromise you have with HPHT synthesis versus CVD.
There is a comment from Pauli Kihaes in response to Fyodor's question. A side comment, Fyodor. I think NVT1 at room temperature is generally independent of P1 density, so it could be getting worse when P1 gets very high. Enter reference. So thank you, Pauli.
Yeah, I think that basically they detune it, right? So in the sense that the direct flip-flops are in a high field, not very probable. But on the other hand, they can be more complex processes. Like if you have very high concentration of NV centers, then two NV centers can make a flip-flop, and then somehow make a flip of P1. But again, it depends on the resonance between them. So I'm not 100% sure. It can be some causes of kind of decoherence or relaxation, also more complex than direct flip-flops between spins.
And I don't know if my comment here will be correct, but another follow-up effect of T1, like perhaps in these particles, if you have some like defect site that involved in like phonon scattering, that could, because in general, it's like the T1 relaxation is the exchange of energy, the loss of energy back to the lattice after you've excited the spin system. So, if you have like maybe a higher concentration of defects, and this is more related to the crystal quality probably as well. So if you have maybe some defect scatterers or something like that, that could affect this energy exchange with the lattice, which would affect T1. Mm-hmm.
So kind of a summary of this work is that it seems around 20 ppm of nitrogen currently seems optimal in HPHT particles. It is possible to be produced at grams level. We have some plans to launch a new product with lower nitrogen content. So this was kind of very good research with some practical outcomes coming from it. If there are no more questions, probably we will wrap up today's conference because we run it from quite exotic environment. All three of us are at ACS, Regional ACS Conference and Magnetic Resonance Symposium particularly. So there is now lunch break when we were able to run the conference, this virtual lecture, and we invite everybody to join us on November 30th. There will be a talk by Romana Shuragl. She has a lot of very exciting clinically relevant results. So please join us in a month or so. Thank you so much. Bye-bye.
Thank you.
Thank you very much for organizing.